Hello, and welcome to Alcohol and its Effect on Your Brain. We'll be going over neural stem cells and alcohol neuropathology. Let's begin with stem cells. Stem cells are cells that can divide indefinitely and give rise to a variety of cell types. There are several different types of stem cells. Totipotent stem cells, which can produce all cells of an organism. Pluripotent stem cells, which can develop into almost any adult stem cell, and multipotent stem cells, which are more restricted in the types of cells they are capable of becoming. We will focus on multipotent stem cells, as that is where we will find neural stem cells. Neural stem cells are multipotent, self-renewing cells, and under the right conditions, can produce other cell types and give rise to several types of cells in the central nervous system, including neurons and other brain cells. It has been found that these neural stem cells are in all regions of the brain, but suppressed in most. Two main regions, however, show activity of many neural stem cells and are in subventricular zone, or SVZ, in orange, and the dentate gyrus in the hippocampus in blue. Neural stem cells are involved in neurogenesis, which is a process where glia, connective tissue in the brain, and new neurons are created from the stem cells. Neurogenesis is regulated by genetics as well as environmental factors. Interestingly, people with chronic alcoholism show many similarities to the environmental factors that regulates neurogenesis, and it can be determined that alcohol abuse and alcoholism is similar to the regulation of neural stem cells. Alcoholism shows changes in behaviors as well as physical changes in the brain, and given the overlap that alcoholism has with the environmental and genetic factors that regulate neurogenesis, there might be a possibility of even treating or preventing alcoholism in the future. Research has been done in the past showing alcoholism disrupts the formation of new brain cells in a person that is developing, but we are going to focus on adults and how it affects their brains. Let's start with genetics. Studies have shown that genetics plays a significant role in the risk of developing alcohol dependence and excessive alcohol consumption habits, which includes sensitivity to alcohol intoxication, alcohol withdrawal symptoms, and the preference for alcohol over other drinks, such as water. Genetics influences three main components of neurogenesis: neurostem cell proliferation, cell survival, and cell differentiation. A study with mice was done where different strains of mice experienced differences in these functions. In one strain, the neural stem cell proliferation was the highest, while another experienced cells that turned into neurons. This suggests there is a significant difference in genetics for how alcohol affects the brain. Next, let's talk about environmental factors. In a similar test as before with the mice, it was shown that an enriched environment that promotes activity and learning had a significant increase in neurogenesis compared to mice in standard living conditions. This is because exercise and learning increase neural stem cell survival and differentiation. Another important aspect of environmental factors is stress levels. Stress reduces neurogenesis, and in some cases induces drinking, which is significant as it plays a role in both neurogenesis and addiction. Now let's transition into the mechanisms involved in alcohol use and neural stem cell regulation. Alcohol alters a variety of neurotransmitter receptors and signals, including glutamate and serotonin, which influence neural stem cell proliferation in adults. This is through actions on chemicals that bind to neurons responsible for nerve signaling. The second mechanism is glutamatergic neurostem cell regulation, which is thought to inhibit neural stem cell proliferation by downstream effects on a particular type of glutamate receptor through an indirect pathway. The third mechanism is serotonergic stem cell regulation. There is a correlation between conditions that decrease the number of serotonin neurons and factors that decrease neurogenesis, which suggests that this neurotransmitter is important in the regulation of adult neurostem cells. Inhibiting serotonin synthesis decreases neurostem cell proliferation in both the SVZ and dentate gyrus. The last mechanism of regulation of neurostem cells is by growth factors. Growth factors bind to neurons and regulate neurogenesis at every stage: proliferation, differentiation, and survival. Alcohol disrupts adult neurogenesis by affecting secretion of growth factors, leading to changes throughout the process. Now let's look at the damaging effects alcohol has on the brain when used in excess. Some common side effects of drinking alcohol include difficulty walking, blurred vision, slurred speech, slowed reaction time, impaired memory, and many more. Some of these quickly stop when drinking stops, but on the other hand, a person who drinks heavily over a long period of time may have brain deficits that last longer after drinking. It is a heavily researched topic, and how these effects can be reserved and prevented in the first place. Some of the factors that affect the extent of alcohol in the brain include how often a person drinks, the age when someone first starts drinking, how long they have been drinking, the person's gender, genetic background, and family history of alcoholism, as well as his or her general health. Memory loss is common after a night of drinking. The more intoxicated a person gets, the more likely they are to black out or not remember events from the previous night. During a blackout, alcohol is interfering with the receptors in the hippocampus, which prevents them from communicating properly. When this happens, long-term potentiation is disrupted and results in temporary anterograde amnesia. Long-term potentiation is also very important in memory and learning. Thank you for watching, and hopefully you've learned more on alcohol and its effect on the brain.